So before beginning into some of my research, I'd like to give a brief summary of my background. So I'm from Penn State University. I received my PhD in 2008, after which I joined the research and academic faculty at Penn State. I'm currently the head of computational methods development at the Applied Research Laboratory, and also have an appointment as assistant professor of mechanical engineering. And I'm also in the graduate faculty in bioengineering. I do a lot of bioengineering research that you'll see. I advise students in both of those departments, so I'm advising or co-advising 4 PhD, a couple master's students, and a couple of undergraduate honors students. So if you're not familiar with the Penn State Applied Research Laboratory, just very briefly, Penn State ARL was established in 1945 by the U.S. Navy. It was at the time and remains today one of the premier hydrodynamics research organizations for the U.S. Navy. We also do a lot of other manufacturing in addition to hydrodynamics right now. Officially, the lab is a Department of Defense university-affiliated research center. There are 5 such centers across the U.S. For example, the Applied Physics Lab at Johns Hopkins is another institution. We have over 1,000 research faculty and staff at the laboratory. And one thing that's really unique about Penn State ARL is that we have a very close relationship with the university. We're on the campus, and a handful of us also have appointments in academic departments. Myself and one of my colleagues have appointments in mechanical engineering. There are a couple in aerospace engineering, bioengineering. So my particular background is my group is in the computational mechanics division at the ARL, at the Applied Research Lab, where there are 3 departments. Computational methods development, which I lead, the advanced fluid thermal modeling, and CFD analysis. So really these 3 departments constitute a very unique combination that I haven't found anywhere else in software development, basic research, and applied research. So we do everything from developing computational fluid dynamics and structural mechanics software to actually applying that software. For example, the CFD analysis group does design and analysis of real Navy hardware that's being deployed in the fleet. We extensively use high-performance computing, so we have 4 on-site clusters with about 8,000 compute cores, and when we need more than that, we have access to the Department of Defense high-performance computing program. For example, one of the clusters that recently deployed this June was number 14 in the top 500 list that I'm using a lot lately. So my technical expertise is in computational fluid dynamics. Multiphysics modeling is my background. I do a lot of fluid structure interaction. Coupled CFD6 software, do a lot of turbulence modeling, large eddy simulation. I also have a lot of experience in medical image processing and 3D anatomical reconstruction. You'll see some of the patient-specific modeling that I'm doing. And all of this requires high-performance computing. So I apply those capabilities in a few different application areas where my real research objective is to use computational modeling and high-performance computing to solve very challenging problems in a range of different areas. One of those is in biological fluid dynamics. So I'm doing a lot of basic science research in biological fluid dynamics. For example, I'm going to show you today how we're trying to figure out why the dog, canines, have such a keen sense of smell. And if we can figure that out, then we can design bio-inspired devices to outperform current technology for chemical trace detection. I'm also looking at, we're trying to figure out now, does anything change underwater? So how has nature solved the problem of detecting chemicals underwater? So we're working on that problem that I'll show you today. I also do work in biomedical engineering. I'll show you some work in cardiovascular devices, and I've gotten into work in the respiratory side as well. In the past few years, I've been doing a lot of work in renewable energy, in particular wind energy. And by virtue of the laboratory that I'm affiliated with, I do also some naval hydrodynamics research. So this particular project is something that began with my PhD dissertation, where we're basically trying to reverse engineer the dog's nose. So it's known that a canine has an extremely acute sense of smell. They can detect chemicals on the order of about 1 part per trillion, which is 10 to 100,000 times our sense of smell. So we're trying to figure out why dogs have such a keen sense of smell. And if we can figure that out, then we can design bio-inspired devices to improve current chemical trace detection technology. So how do we do that? Well, we start with cadaver specimens, and we use high-resolution medical imaging, CT or MRI scanning, to acquire raw image data that we then use in-house and commercial software to process those data and basically extract the anatomical information, which we then basically stitch together using algorithms into a 3-dimensional model that we can use for numerical simulations. So the first thing that we found in this work, before we even did any simulations, based on the anatomical reconstruction of the nose, we found that dogs possess a very unique nasal cavity. And what I mean by that is the olfactory region, or the sense of smell in the dog's nose, is located in the rear of the nose. And not only is it located in the rear of the nose, if you take a slice through the middle of the nasal cavity, you can see that it's located in basically what appears to be a dead end region. So this is the front of the nostril. This is the nasopharynx that goes to the lung. So we're functionally trying to figure out how odors get back into this olfactory region where the sense of smell is located, and why on earth would the sense of smell be located so far back in the olfactory, into the nasal cavity? Why wouldn't it be located up front? And this is much, much different than what happens in our nasal cavity. So in our nose, the olfactory region is along the main flow path, along the top of the nasal cavity. And it turns out one of our hypotheses was that this configuration dramatically affects airflow and odor transport in the nose, and therefore olfaction. And what we're finding in current work is that all keen-scented animals appear to possess this architecture. So what we did to study function was we carried out very large, at the time at least, computational fluid dynamics simulations where we solved the incompressible continuity and Navier-Stokes equations to carry out fully transient simulations of canine sniffing. So there's experimental measurements made. Dogs sniff. When they sniff in, and then they expire. They sniff in and out at about 5 times per second. So very fast. So because this geometry is so complicated, we require very large, complex computational models, which at the time, in 2008, when I finished my PhD, these took about 2 weeks to run on 256 CPUs, which at the time was a moderately large computation. Nowadays, we're running on thousands of processors. So what we found in using basically high-performance computing for discovery in this case, what we found is that during inspiration, the dog, once airflow enters the nose, it splits into 2 airflow paths. So there's an olfactory airflow path that I've shown here in red. So these are path lines extracted from the simulation. And that airflow reaches the olfactory region, and all of the remaining airflow goes directly to the lungs. So, and we further found that all of the airflow that makes it to the olfactory region goes through a single airway, which is called the dorsal meatus, or Latin for top airway. And if you follow the olfactory flow path, so the air goes back this one airway, it goes to the rear of the nose, and then the cross-sectional area back there is fairly large, so it decelerates, and it slowly filters forward through all these very complex airways where all the odors are deposited onto the sensory receptors. So during expiration, we found that airflow goes directly from the nasopharynx, which comes from the lungs, out the nostril without ever entering the olfactory region. And this is important because there's basically no airflow in the olfactory region during sniffing, or during the expiratory phase of sniffing. So basically what this means is when a dog is sniffing, every inspiration, there's a large delivery of odorant to the olfactory region, and it's all in a unidirectional pattern. During expiration, there's a time stagnant phase through which odorant can be absorbed. So that's we found to be very important for olfaction. And this is really unique compared to what happens in humans. So we also carried out simulations in humans and found that when a human expires, basically the olfactory region is purged of odor. So an interesting experiment that you can do on your own is to smell something, for example an orange, and you sniff in and you smell the orange, you have the olfactory sensation. But then if you quickly sniff out, you no longer smell the orange. But the dog, in this case, when the dog sniffs out, it continues to have the olfactory sensation. There's odor being absorbed in the olfactory region. And this turns out to be very important. This is also very unique compared to what happens in current artificial nose technology. So that's only... Yes.
But under the equation, if stuff is going into the olfactory region, it has to come out.
Oh.
I don't see any
yes.
going from right to left.
Okay. So it goes in here and comes out at the bottom. So complete.
During the inspiration.
During inspiration. And then it sits there, and then during the next inspiration, it pushes more through. So it actually, I don't have time to go into it, but acts as an accumulation effect. So this is, I have a lot to get through, but I can discuss at the end. This is one of the reasons why air it resides in there for a longer period of time. So it's one of the reasons, ways that dog can have a background odor, so it can detect new odors. There's some regions of the nose back there where There's odors that are residing for multiple sniffs, so there's background that it can compare to. So the airflow is only part of the story. So it's well known that all airways in the respiratory system of mammals are lined with a very thin layer of mucus. So you can see a layer of mucus here over olfactory epithelium. These yellow cells are the olfactory receptor neurons. The olfactory receptor neurons have thin hair-like cilia that project from them. So you can see a micrograph here. Basically, this is a mat of olfactory cilia with receptors. So this olfactory epithelium is lined with a very thin layer of mucus, and this is important for olfaction for one reason, because different vapors are, the solubility of different vapors in that aqueous phase varies dramatically from odorant to odorant. So, and this is all determined by what's known as a partition coefficient. So we looked at 3 different odorants, and this can vary over 6 orders of magnitude. So we simulated the odorant deposition of 3 different odorants. So amyl acetate, which has a smell of bananas. DNT is an explosive that is a derivative of the explosive TNT. Limonene is the orange scent in commercial products. So these are used widely in the olfaction, the field of olfaction. So what we did was we solved a species transport equation for odorant, subject to conservation of mass at the interface and an equilibrium partitioning relationship. And we made some assumptions of steady state, which are valid because the mucus is so thin. And then if we assume diffusion limited binding, we can come up with a relationship, a mixed condition that we can apply to the air phase so that we don't have to solve for transport of the mucus layer. But anyways, at the end of the day, what we have is we found that each of these odorants have dramatically different odorant deposition patterns in the olfactory region. So I'm plotting here the flux of odorant in the olfactory region. So you can see for DNT, which is highly soluble in the aqueous phase, that the odorant deposition is along the front of the olfactory flow path. Amyl acetate, which is moderately soluble, has a gradient across the olfactory epithelium, and limonene is more uniformly distributed. This is important because basically there's a spatial pattern of odorant deposition across the olfactory epithelium. The neurobiology field, in particular Linda Buck, who won the Nobel Prize in 2004 for her work in this area, found that olfactory receptors are spatially distributed throughout the olfactory epithelium. So this combination, and it looks like we're finding, is that the spatial distribution of these olfactory receptors are such that receptors that are sensitive to highly soluble chemicals are located in these regions. Those that are sensitive to more insoluble chemicals are more uniformly distributed. So this is one way that mammals are able to discriminate one odor from another. So it's a patterning. So what we're currently doing is I've extended this work to do a very basic science research collaborative study with a biology professor at UCLA. We're awarded an NSF grant to look at an interspecies comparison of respiration olfaction in different animals. So I'll show you here in a second, but basically we're trying to predict and compare respiratory and olfactory performance in different animals and develop form-function relationships for mammalian respiration and olfaction. So how does anatomical complexity correlate with olfactory and respiratory function? So this particular set of species, we're looking at a lot of different things. So different carnivores versus ungulates versus rodents, different types of structures in the nose. So here's high-resolution MRI scans of a white-tailed deer. You see they have these contorted scrolls of turbinates. These are bony structures in the nose, versus a coyote has this densely branching structure. We're looking at comparing different habitats, predator versus prey, so a number of biological implications, and which has some technology motivation as well. But we're trying to answer some very basic research questions. For example, do some animals have a keen sense of smell, more keen sense of smell than others? We're trying to look at respiratory heat exchange in harsh habitats. So sea otters live in a marine environment and require lots of respiratory heat exchange. So we'll see how that affects how they get that. So we've reconstructed and scanned each of these models. And just to show you what some of them look like, so this is a high-resolution scan of the coyote at 150 micron resolution. So we had to develop basically new MRI scanning protocols to get such high resolution in these specimens. But these are scans from the tip of the nose to the rear of the nasal cavity. So you're in the vestibule now, the front of the nose, and then there's this structure here, a turbinate structure, that begins to branch out and fill the airway, and this is solely to increase the surface area for respiratory heat exchange. There's that one airway that I pointed out before, the dorsal meatus, that goes directly to the olfactory region, and then the nasopharynx goes to the lung, and then this is the olfactory region where odorant deposition and olfaction takes place.
So this is four-dimensional.
This is 3D. It's not changing in time. So to show you how complicated things can actually get, these are high-resolution scans in the sea otter. So the sea otter, because it lives in a marine environment, requires a lot of surface area in its nose for respiratory heat exchange. So you can see how incredibly complicated it can get. So what we're trying to do is we're trying to simulate airflow, odorant deposition, heat exchange in these animals using computational fluid dynamics and high-performance computing. So because these models are so anatomically complex, the computational models are quite large, so on the order of 10 to hundreds of millions of computational elements. So this requires upwards of thousands of cores on a high-performance computing cluster to simulate this. And you require such fine meshes or computational models because you're modeling mass transfer in an interface where there's very large gradients. So you require such large, complicated models. We do a lot of parallel post-processing, visualization, and rendering. I'll show you here in a second. But at the end of the day, the goal of this project is to compare form and function in different animals. So nasal airflow patterns, respiratory heat exchange, and odorant deposition. So here's an example of some of the visualization we can do to illustrate what the flow looks like. So here's an example of a computational fluid dynamics streamline that we extracted from our solution. What we're going to do here is basically animate the camera through the nose such that it's basically a virtual endoscope. So we can look at what the flow looks like going through the nostril. So the flow getting pulled into the nose is potential flow, so it's very slow. As you go into the nasal cavity where the cross-sectional area is reduced, you go very quickly, and we're following this streamline right here. So in that region, the cross-sectional area expands, so you're starting to slow down. If you look, as you come out of the respiratory region, you're going directly to the nasopharynx, you can see the olfactory region back here. So it's kind of like a virtual endoscope. But at the end of the day, we have these comparisons of airflow, odorant deposition, respiratory heat exchange in each of these animals. So what we're doing now is looking at, is anything different in water? So we're going to compare chemical sensing in air versus water. And the application of this, there are a number of naval applications that are interested in detecting underwater chemicals. So we're looking at how fish have done it. So to do this, sharks are known to have a very keen sense of smell. In particular, the hammerhead shark is thought to have several advantages. So what we're doing is and I have a PhD student that's just wrapping up his thesis right now and will be defending it here soon, but we've reconstructed a three-dimensional model of the hammerhead shark and are carrying out CFD simulations of water flow and odor deposition. So when you look at the anatomical model, the hammerhead shark has what's called a cephalofoil. And on each side of the cephalofoil is an olfactory chamber where odorant sensing takes place. So when you look at this, there's an inlet to the olfactory chamber right here and an outlet underneath the head. When you look inside, the structure is much, much different than what you see in animals. So it's comprised of several rows of olfactory lamellae, which are just thin connective tissue lined with olfactory epithelium. So we've generated computational models and are now carrying out very large-scale simulations to understand the basic science of this problem. So what we found that the head morphology of the hammerhead shark is very unique in that it drives flow through the olfactory chamber. What happens is the inlet to this olfactory chamber is located at the front of the cephalofoil where the stagnation pressure is very high. The outlet is located around the bottom side of the head where the flow is accelerating around this foil shape, and we know from fluid dynamics when the flow accelerates, the pressure decreases. So there's a high pressure at the inlet, a low pressure at the outlet, which drives flow through it. So basically it's a ram effect. And from a bio-inspired point of view, this is important because it means we could actually use this mechanism as a means to drive flow through an artificial device rather than relying on a pump. So we've also simulated flow distributions inside the chamber, so you can see how flow comes in this inlet nostril around and how it filters through these olfactory lamellae. So we're also interested in comparing, looking at the odorant deposition. So it's well known that from the fish biology literature that fish also have a thin layer of olfactory mucus that lines their airway. Unfortunately, the partition coefficient between seawater and olfactory mucus is unknown. So I collaborated with Adri Van Duyn, a mechanical engineer at Penn State, and we carried out molecular dynamic simulations of odorant partitioning between seawater and a simplified phase of olfactory mucus. And these were on one of our high-performance computing clusters at Penn State. So in what we found, we simulated 4 different odorants. So fish are known to detect amino acid odorants in water. And in particular, sharks are known to, these odorants are known to cause, to stimulate feeding. So we looked at 4 different odorants that span response thresholds for sharks reported in the literature. So in what we found, that in each case, all of these chemicals throughout the nasal cavity, you get very, very similar deposition patterns. So what's happening in fish is because fish live in an aquatic environment, there is not this large spatial deposition pattern as there is in mammals. And this is because we found that the odorant partition coefficients have a limited span, so they only span about 1 order of magnitude. And this is important for 2 reasons. One, from a fish biology point of view, people have wondered for years why the olfactory epithelium of fish is much, much different than it is in mammals. In that, there's no spatial organization of the olfactory receptors in the epithelium of fish. It turns out we find that fish could not even utilize such a spatial distribution of olfactory receptors because there's a uniform distribution of odorant. So it wouldn't even be utilized. So that's probably why fish have adapted or evolved to not have that. So in the biological fluid dynamics realm, I have several initiatives that I want to pursue in the future. I really would like to start pursuing transitioning some of this technology, and I've already begun that. I've formed a collaboration with colleagues at NIST, so in trying to incorporate some of these biomimetic principles into artificial chemical sensing. So here's an example of a preliminary study I just worked on with a colleague at NIST, where basically he designed an end cap such that it functions, functionally at least, like the aerodynamics outside of the dog's nose. So I didn't have time to show you. When a dog sniffs outside of its nostril from our CFD simulations, when the dog sniffs in, odorant is pulled in directly in front of the nostril. And when the dog sniffs out, the configuration of the nose is such that there's jets vectored to the side. So when my colleague here designed an end cap such that there are jets vectored to the side and it sniffs in through the center, he found that current detection technology can significantly improve performance. He can extend the range over which these chemical detectors can pull in odorant. I'm also looking at extending this work into other areas. So I think there's a lot of opportunity for interdisciplinary research with biologists. So an example is a preliminary study I did with a professor of biology at University of Utah, where it's well known that there's unidirectional flow in bird lungs. What we found, she did in vivo experiments, and I did simulations based on her experiments. These were pretty large-scale simulations. We found that also in iguanas, reptiles, there's a uniform directional flow pattern that sets up over the gas exchange surfaces. So there's a number of evolutionary implications that we're exploring with that. This is prescribed mesh motion. So I developed a custom boundary motion, basically solver. There's no fluid structure interaction because she measured the motion of the lungs and I employed it. And I'm also transitioning this basic research into the field of biomedical engineering.
So basically prescribed motion.
Prescribed motion, yes. But in an attempt to match the experimental motion, yeah. So in the field of biomedical engineering, I have a couple projects in the area of cardiovascular devices. This particular device that I'll show is a collaboration with a physician at the Hershey Medical Center, Frank Lynch, an experimental bioengineer at Penn State, Keith Manning, and my colleague, ARL in mechanical engineering, Rob Campbell, who is in structural mechanics. So this particular project, we're looking at inferior vena cava blood clot filters. So there's a condition known as pulmonary embolism, which you might have heard of, where you get a blood clot that will form in your leg, and it can be transported through the iliac vein in your leg, up through a large vein in your abdomen, and which returns blood flow to the heart. And if the blood flow makes it to the heart, it goes to the lungs. And if a blood clot makes it to your lungs, it can be fatal. So there are about 1 million instances of pulmonary embolism in the U.S. each year. About 1/3 of those are fatal. So what they often do is administer anticoagulation medication, which oftentimes doesn't work. So what physicians will have to do is deploy these filters. So what they do is here's just an animation showing you the inferior vena cava. This is an anatomical reconstruction, is located in the abdomen. And then they deploy a filter to capture a blood clot if a patient is at risk for having a blood clot. And then they can be, when the patient is no longer at risk, they can be removed. So they just go in through a catheter. So the objective of this study is to develop a computational method that we can use to predict the performance of these IVC filters, because there's a number of problems clinically.
What's the typical size
Of this vein, a couple centimeters. Yep. That may be used for patient-specific selection and placement of these filters. So what we do is we begin with patient CT data, reconstruct an anatomical model, and then we construct a volume model of the vein wall. And in this particular study, we were using ex vivo properties, material properties of veins measured in cadavers. Because we're doing that, we require the use of an inverse analysis that I'll briefly describe here in a second to obtain the in vivo stress state of the vein. And that's important because when we deploy this filter, as they do in the surgery, so we have a sheath, they go in through a catheter, this filter's in a sheath, and they deploy it, and it comes into contact with the vein walls. We're using finite element to model that contact and the deployment of the filter. So when we have that configuration, then we carry out computational fluid dynamics simulations to look at what effect the blood clot has on the flow. What happens if a blood clot is trapped in the filter? How does that affect the blood flow?
So basically the motion is described by experiment.
Mm-hmm.
And you do that dimension.
Yes. Yes. So in this particular study that was we just published in Journal of Biomechanical Engineering, we had 2 patient veins, and we used nonlinear structural modeling for both the IVC filter, which is nitinol, and the vein, where we used an anisotropic hyperelastic tissue. And we used ex vivo material properties from a cadaver vein, and these material properties were measured in an ex vivo, they were cut from a cadaver. So to do that, we had to carry out an inverse analysis to be able to use those data, where we started with a reference configuration, and then we deflated the vein and compressed it axially by 15%, which was what was measured in that experimental study that measured the properties. And then we inflated the vein using the ex vivo material properties and applied and then compared it to the in vivo anatomy and took the difference between the two, adjusted the reference configuration, and iterated until we obtained the exact in vivo geometry, except this case had the approximate in vivo stress state. So in this geometry, and then we used nonlinear contact modeling to model deployment of the filter. So, and then we carried out CFD simulations to look at the patient's specific hemodynamics. So I'm just going to illustrate a few interesting things that we found. So in this particular patient, which has an anomalous IVC, we found that this patient is at risk for forming a blood clot in the inferior vena cava. And this is because there's large recirculating flow in that part of the inferior vena cava, and in the cardiovascular system, recirculating stagnant flow is a bad thing. It's typically where blood clots can form. So this actually corresponded to some studies in the literature that have shown that this type of an IVC anomaly, these patients have a high instances of forming pulmonary embolism. So this might be one of the reasons why you get large stagnant flow regions in the inferior vena cava. And then when we deployed the filter and put in a notional clot that was based on a clinical study, you can see there's an even larger flow recirculation region, which means that this patient were actually to get a clot captured in the filter, that clot would likely grow and basically then, in the end, occlude the vein. So I have a few research directions that I want to pursue in biomedical engineering. One is patient-specific modeling, such as I've shown here. The next step of this work is to use fluid structure interaction to start looking at blood clot migration through the circulatory system and how these clots are captured by these filters. I've also been collaborating recently with the Artificial Organs Group at Penn State Hershey Medical Center, where we are looking at artificial hearts and blood pumps. So Penn State Hershey is one of the pioneers in the development of artificial hearts. It had one of the first artificial hearts on the market. And I'm collaborating with this group right now, and we recently got a preliminary NSF or NIH award to get preliminary data for an R01 that we're going to be submitting this summer. But basically, we're developing new methods for designing artificial hearts. And one of the components of this is high-performance computing is going to be critical, because what we're proposing to do is one of the difficulties in designing artificial hearts is that it's very difficult to do without damaging the blood. So if you have a lot of shear stress in the flow, you can damage the red blood cells. So when you do that, that's known as hemolysis. So the current capability of predicting hemolysis is quite poor. So what we're doing right now, I just started this the past few weeks, is we're developing a much better and improved hemolysis model where we're using large eddy simulation and very large high-performance computing models where we can get much better predictions of hemolysis. And we're doing joint experiments to validate these models. So on the respiratory side, I've begun collaborating. I'm trying to push my olfaction work into NIH. And I began collaborating with a neurobiology professor at Yale who's done a lot of work in NIH olfaction-related area. And we're looking at using multi-scale modeling to better predict olfaction. So there's a recent PNS study a few years ago that did a nice job of predicting the olfactory response of olfactory receptor neurons. And I'd like to couple this with computational fluid dynamics for a multi-scale approach to olfaction. I also think there's a big opportunity to do patient-specific modeling of respiratory functions. So in the field of like otolaryngology or pediatric airway disorders. So I think there's a lot of opportunity to reconstruct patient models and use those for pre-surgical assessment that surgeons can use to guide their surgery. So in the area of renewable energy, I've been doing a lot of work in the past couple years in wind energy. So I'm just going to summarize some of that work. So this is part of the Penn State Cyber Wind Facility that's funded by the Department of Energy. So this is led by Professor Jim Brasser in mechanical engineering, and I'm contributing in the ways that I'll show you. But the objective of this is to develop a high-performance computing cyber wind facility. So this is extremely large-scale simulations, and I'll show you why this is required. So we're simulating the wind turbine, platform, wake interactions with the atmosphere and ocean. So basically, this is geared towards offshore wind turbines. So, and this is cyber wind facility really means that we are using mesoscale weather predictions coupled with atmospheric boundary layer simulations of the turbulent eddies in the atmospheric boundary layer, coupled with the computational fluid dynamics simulations of flow around the wind turbine blade, I'll show you here in a second, and coupling that with the 6-DOF motion of how wind turbine platforms respond and how that affects the performance. And this is important because we can use this for basic research in wind energy, and also designers of wind turbines can use this as basically a very detailed data set like they would for experiments, because experimental data for all of these details is not available and very difficult to obtain. So I'm contributing this work in two ways. So I'm doing a lot of very large-scale computational fluid dynamics simulations on high-performance computing clusters. So to give you an example of how extremely complicated this geometry is, so these meshes are typically on the order of hundreds of millions of cells. This particular one is 150 million cells. We're doing large eddy simulation of the atmospheric boundary layer, which is Jim Brasser's specialty. So this is a large eddy simulation of the atmospheric boundary layer. These red flow structures are large turbulent eddies that are turning over in the atmosphere. So our hypothesis is, and this seems to be the case, is that these large-scale turbulent eddies in the atmosphere significantly influence the performance of the wind turbine because they change the incoming angle of attack very quickly of that wind turbine blade. So the blade will stall and you lose lift on the blade, and it becomes basically an unsteady lift force that generates variable torque on all the components and reduces the lifespan. So these are large simulations that we're carrying out on about 10,000 cores on the new supercomputer at Texas Stampede, which I think is number 6 in the top 500. But our preliminary results, and these are actually being presented today at the AIAA meeting, is that we're finding when you compare uniform inflow, which is basically what every other group is doing right now, basically the flow looks nice and attached. These are surface limited streamlines, so it's showing what the flow looks like over the wind turbine blade. Most of the flow is attached except at the root. That's known to cause some separated flow. But when you start to look at introducing the atmospheric boundary layer, what seems to be happening in the real system is that you get these large regions of separated flow that vary in time as these turbulent eddies pass through the wind turbine rotor. So and that significantly affects the performance, not only the performance of the wind turbine, but varying loads which reduce the lifespan. So the gearbox is known to fail a lot.
So the subjects do 60 degrees of freedom.
Yeah.
It's like moving more or less.
For the wind turbine blades, they're rotating. So these are all rated for they all try to turn at the same RPM constantly. So they have a control system that tries to keep them at the same RPM. So and where I'm contributing a lot more, actually, is I have a PhD student working in this area, is now trying to predict the platform dynamics of these wind turbine platforms. The current state of the art when they design these wind turbine platforms is reduced order models. So they have reduced order models that they use to design this. They don't use higher fidelity simulations. So what we're trying to do is to develop and validate a coupled CFD on 6-DOF approach to be able to simulate these platform motions.
So how do you find how you come?
I'll describe that on the next slide. Oh. So what we've done is we've developed a partition, or known as sequentially staggered coupled algorithm. And tightly coupled, I mean basically what we're doing is we have a subiteration inside of our time loop where we're subiterating on the 6-DOF equations of motion and the Navier-Stokes equations until they converge. And if you do that in an intelligently fashion, and you have to do this actually for these types of problems, and I'm sure Professor Lewis has worked in this area, loosely coupled approaches don't work. For large motions, certain density ratios, the schemes are unstable. So if you do this in an intelligent fashion using dynamic relaxation, you only need typically a few subiterations. In some cases with really high acceleration on the order of 5 or so, but that's it. So this is implemented in an open source package, OpenFOAM, which most of this work that I've shown you today uses. And it's been validated against benchmark experiments. So here's an example of historical benchmark data of a free heave of a circular cylinder that we validated and compared quite nicely with compared to experimental data. So now we're using that capability to start to simulate these offshore wind turbine platform dynamics. So here's an example of a simulation my student just carried out for the rolling motion of a platform, so how it damps out. So these are very recent experimental data that were required for this platform. And you can see for free heave, you can see we're matching the experimental data quite well. So in the future, we're going to be implementing nonlinear mooring line restraint models. So these things are moored to the seabed. So we're going to be implementing a mooring line model and then start to implement realistic ocean wave conditions via wave buoy data. So finally, I just wanted to wrap up, show you 2 slides on some of the naval hydrodynamics work I've been working on recently, just to illustrate some of the computational capability. So this is an area that's sponsored by DARPA, where the objective is to develop a HPC, high performance computing, FSI capability for simulating very high Reynolds number naval applications. So, and DARPA is funding us to do our experimental group did benchmark experiments, and then we've developed a FSI capability to simulate this problem in an HPC environment. So the benchmark experiment is a circular cylinder. And from fluid dynamics, you know downstream of a circular cylinder, there's vortex shedding. And that vortex shedding buffets the hydrofoil. So this is a flexible hydrofoil. And then the flexible hydrofoil deforms. So what we did was we used FSI to design the experiment before the experiment was even run. And the experiment turned out quite well, just using coarse models. So these are a few million cells. And now we're carrying out higher fidelity large-eddy simulations for validation with the experimental data, because this is a turbulent flow problem. This is a pretty large model in the order of about 40 million computational elements. They're running on the Spirit cluster at DoD HPC, so about 1,000 cores. So what I've tried to do is to show you how I use computational modeling and high-performance computing to solve pretty challenging problems in a number of different areas of basic science research and also in a few application areas. I use a lot of open source software. For example, OpenFOAM, which is an open source computational continuum mechanics library that I've been using for a number of years now. Python, ParaView, a number of open source packages on HPC systems. This is a real advantage in my opinion, especially when it comes to a few funding agencies. So for example, NIH requires model sharing in most all NIH grants now. So you have to be able to share your models. So, and we are able to do that. Department of Energy, some of their newer renewable energy research, you have to have open source or distributable software. So, and that's never a problem for us, whereas some people it is a problem for. I do a lot of collaborative and interdisciplinary research. So I collaborate a lot with biologists, physicians, also other engineers, so structural mechanics. My background is mostly fluid thermal science, but for example, in the blood clot filter modeling, my structural mechanics colleague was able to handle and advise our student on all of the structural mechanics aspects of contact modeling. So basically, I do a lot of collaborative study where I collaborate with different engineers in different fields to solve problems that neither one of us could independently. So I have a lot of experience pursuing funding and securing funding from National Science Foundation, NIH, DARPA, and I have a number of students that I'm advising. And apparently, and that's... I have 3 minutes, but that was my question slide.